Hello, everyone. I'm glad to present the research submitted to iMeta from the Fan Xiaohui group of Jingyang University, titled A Comprehensive Benchmarking for Spatially Resolved Transcriptomics Clustering Methods Across Variable Technologies, Organs, and Replicate. To begin with, I would like to briefly introduce the background of this study. Spatial clustering is a crucial step in the analysis of spatially resolved transcriptomics, SRT, serving as the foundation for downstream investigation of tissue heterogeneity. Although many computational tools have been developed, selecting the most suitable method for real applications is still a challenge. Here, we benchmarked 14 spatial clustering methods with nearly 600 datasets, including both real and simulated data. We provided practical recommendations for selecting methods based on different technologies, organs, and biological replicates, involving cell type clustering and spatial domain identification. We assessed the influence of data characteristics and spatial patterns on clustering accuracy, and offered the optimal pre-processing pipeline for spatial clustering methods. Our datasets came from publicly available resources, with expert manual annotation or previously validated clustering results as ground truth. First, we analyzed brain datasets from 10 classic and widely used platforms. Method performance varied by platform with different spatial resolutions, which also dictates the nature of clustering tasks. ST and 10 times Visium aggregate transcripts from multiple cells, constraining annotations to spatial domains. These platforms are better suited for methods like Stagate and GraphST. In contrast, high-resolution platforms such as StereoSeq and Xenium enable single-cell resolution, where cell-type clustering becomes important, and benefit from methods like ST-Learn. Notably, Base and Banksy demonstrated flexibility in balancing both tasks effectively. Then, to evaluate how organ-specific architecture affects clustering accuracy, we analyzed datasets generated with the 10 times Visium platform across organs. We quantified the spatial characteristics of domain distributions, and found that method performance is closely related to the spatial continuity of organs. For example, the cortex displayed well-defined layered structures with high spatial continuity, which favored methods like Stagate and SEDR. In contrast, methods like Precast and ST-Learn performed better on organs with lower spatial continuity, like the lung and liver. These performance differences are also related to the spatial continuity assumptions of the methods. When analyzing more complex datasets like SlideSeq, which captures more disordered cell-type distribution patterns, performance dropped due to the lower spatial continuity. SRT data often show variation even between adjacent tissue sections from the same biological sample. To test robustness, we used SC-Cube to simulate biological replicates on 10x Visium and Murfish datasets. On 10 times Visium data, DeepST and Banksy achieved the highest accuracy, while on datasets, Base and ST-Learn outperformed others. Then, we simulated spatial neighborhood disruptions by merging two adjacent clusters and adding new clusters. Methods like DeepST, SEDR, and Stagate exhibit greater resilience to shifts in domain boundaries and perturbations of spatial neighborhoods. Given the variability observed across technologies and organs, we next explored how intrinsic spatial patterns impact clustering accuracy. High sparsity reduced clustering accuracy for most methods. The number and distribution of clusters also affected accuracy, which was influenced by the definition of spatial neighborhoods and the integration of expression profiles with spatial neighborhoods. We also tested the effects of pre-processing steps on clustering accuracy and identified the optimal pipeline for each method. In conclusion, we systematically compared 14 clustering methods across different technologies, organs, and biological replicates, and provided recommendations for method selection. Our study showed that performance variability is largely driven by dataset characteristics and methodological choices, including sparsity and distribution. We also highlighted the crucial role of pre-processing steps in enhancing clustering accuracy. We uploaded the simulated datasets, method recommendations, and optimized pre-processing pipelines to GitHub for public access. Thank you.